Despite the billions of people spread across our planet and the extreme diversity in physical appearance and culture, science has shown without doubt that our species originated in Africa. And we are all descended from a population of Africans that lived sometime between 200,000 and 100,000 years ago. At that time, we lived as hunter-gatherers with no domestic animals or plants, and small social groups at relatively low population densities. Today, most humans rely on domestic plants and animals for our food and live in dense concentrations in cities, increasingly interconnected by technology. Despite tens of thousands of years and huge economic differences, those progenitors 100,000 years ago were a lot like us. They were modern humans. And while all species are unique, modern humans are an exceptional outlier in many ways, falling way outside the range of variation of other species. How we got to be this outlier species, and the scientific evidence that fleshes out our understanding of that story, is the subject of the next two presentations. In the next two presentations, we'll provide a brief overview of what paleoanthropology has learned about the origins of our unique species. Paleoanthropology is the scientific study of human origins. Any scientist conducting research relevant to our understanding of human origins is doing paleoanthropology. Archaeologists doing paleoanthropology typically use the material record of tools and food remains to reveal the life ways of our ancestors. Physical anthropologists study the skeletal remains of the fossil humans, while evolutionary geneticists use the modern and fossil genome to better understand human evolution. Other scientists join in as well. Climate scientists, geologists, computer modelers all contribute to the very transdisciplinary science of paleoanthropology. Paleoanthropologists often focus on documenting and explaining the origin of key adaptations in our species and its associated lineage. Adaptations are anatomical or behavioral or cultural adjustments to environmental conditions. We seek to understand the adaptive system of humans through time, and an adaptive system is the complex of related sets of individual adaptations. A general trend in human evolution has been the increasing significance of culture in the overall adaptive system, while other non-human animals lack that cultural adaptation. Biological adaptations are those most typically expressed in anatomy and physiology. Examples include the long, pointed teeth of saber-toothed cats, specially designed by natural selection to help kill large prey, and the long neck of the giraffe, molded by natural selection to reach the high leaves and trees on the grasslands of Africa. Biological adaptations can be beneficial in that they provide a raw material advantage for an animal. That neck of the giraffe means it can always reach those leaves. But biological adaptations also impose constraints. That long neck reduces the speed a giraffe can turn around and face a predator. And the transmission of biological adaptations is strictly genetic, and thus slow and constrained by sex. Animals also have behavioral adaptations. For example, antelope in Africa have something called a flight distance. This is the distance to a predator at which point they will run away. This is instinctual and it's embedded in their genome. But some behavioral adaptations are learned as well, so transmission can be both genetic and learned. Behavioral adaptations are more flexible than biological ones, but they can also be lost and then need to be rediscovered. Cultural adaptations are always learned, and they are commonly taught through the use of symbolic references. Anything taught with language is part of culture. The amazing aspect of culture is it is cumulative. The complexity of what is taught can get greater and greater through time, allowing, for example, technologies to become extremely complex. The transmission is always through teaching and normally by use of language and repeated imitation. Humans have a unique adaptive system where culture is the dominant part. We want to know when, where, how, and why this evolved. The foundations of this unique cultural system of adaptation are humanity's complex cognition and extreme prosociality. These two traits make human culture and society possible. Our advanced cognition, made possible by both a very large brain and its complex wiring, allows many important functions. These include the following. Enormous data storage. This allows retention of information such as rules, norms, and recipes for tool manufacture. Extreme creativity. This allows novel advances and insights. Symbolic thought and language. This allows communication and information storage outside the mind. Advanced quantitative skills. This allows people to calculate odds in crucial decisions. Shared intentionality. This is the ability to know that I know, that you know, that I know. This underlies our ability to understand the motivations of others, which helps us be social. An enhanced working memory capacity powering executive function. This brings all that advanced cognition together as a functioning system. The extreme prosociality of modern humans is the proclivity to cooperate at high levels with non-kin. Other animals cooperate with kin, and this is easily explainable through standard natural selection theory, but rarely do they cooperate with non-kin. Humans do it all the time in very complex and repetitive ways. In a talk I attended by Sarah Hrdy, the famous primatologist, she asked us to imagine 200 chimps lining up to get on an airplane, sitting in seats for many hours, and then calmly getting off. It would never happen. Cooperative breeding is an expression of prosociality and includes behaviors such as alloparenting and formal mate exchange. Our prosocial behaviors also express themselves in our tendencies to follow rules and enforce third-party punishment. There is a scientific consensus that both prosociality and correct cognition are embedded in the genome, universally shared by all modern humans, and are key foundations to our unique modern adaptive system. When, where, and why did they evolve? How do we examine the paleoanthropological record for evidence of when these traits begin to evolve? We use proxies. Some proxies are direct while others are relatively indirect. For example, the development of large brains is a pretty direct proxy for an evolving cognition, and the appearance of artwork and symbols is as well, but most proxies are indirect. For example, complex tools indicate increasing amounts of learned behavior, and long-distance movement of material culture suggests extended social networks. There are many other proxies, so let's now examine the record and see when and where these traits start to develop. The genetic and anatomical record clearly shows that our closest living relatives are the two species of chimpanzees, the common chimp and the pygmy chimp, the latter more properly known as the bonobo. This record shows us gorillas branched off from a lineage shared with the chimps and us, and then sometime between 8 and 6 million years ago, our lineage, the hominid lineage, separated from chimps. The hominid lineage is indicated here by the species shaded in gray to the left. The names indicate species recognized from the fossil record, and the red lines are the time spans we recognize from those species. All hominins share a key trait, bipedality, or habitual upright walking on two limbs. Chimps and gorillas are quadrupeds. They walk on four limbs with knuckle walking. As you can see from this close-up of the prior graph, between 8 and 5 million years ago, there are few fossil species. This is likely due to poor recovery. We simply have not found many fossils and sediments of the correct age. But even at this early stage, we see some evidence for that key defining anatomical trait of our lineage, bipedality. The earliest known biped and hominin is from a surprising location, in the modern country of Chad in the Sahara. This is surprising because the vast majority of early hominin fossils have found in eastern and southern Africa. This hominin lived in this location when it was much more lush, and there are good indicators of a moist, wooded environment. There are no postcrania found with this specimen, but there is a well-preserved cranium. The foramen magnum, the hole in the skull where the spinal cord enters, is positioned near the base of the skull, suggesting the head was held upright, and this is consistent with bipedal locomotion. Another early fossil is a femur bone found in Kenya and dated to about 6 million years ago. The femur is important because a biped hominin has a specific shape. This specimen resembles a biped and not a quadruped. A third fossil taxa that dates to between 6 and 5 million years ago, Ardipithecus cadaba, is a clear biped, so we are confident that by 6 to 5 million years ago, there are bipedal hominins present, and these are at the base of our lineage. Bipedality is a defining feature of our lineage, but it is unclear if this trait relates directly or at all to those two features that make humans unique, a complex cognition and prosociality. This graph depicts the state of the evidence in a slightly different manner. Please study it carefully. It shows that after we find these two earliest bipeds, we then have a series of fossil taxa that date between 6 and 4 million years ago that we assign to the genus Ardipithecus. Ardipithecus is best represented by a spectacular set of fossils assigned to the species Ardipithecus ramidus. On the left-hand side of this figure are the bones of two individuals laid out to display their completeness. On the right is the famous fossil named Lucy. This is a partially complete skeleton of a single young female. On the left is the newly discovered Artie. On the far right is an artist's reconstruction of Artie. Artie clearly is a biped and has an interesting mix of features. Notice the long fingers and the long toes. Ardipithecus ramidus is found in Ethiopia in the fossil-rich Afar region and dates to about 4.2 million years ago. The body size is quite small, roughly the size of modern chimps, and some individuals even smaller. The brain size is the same as a chimp, but it is clearly bipedal. It has long arms and fingers, and this suggests it was an excellent climber. A surprise is the funny feet. They are long with a splayed big toe. These are not what we would expect for the feet of a biped. The scientists who discovered Artie suggest that this species walked upright on large tree branches, not on the ground, as we expected the first bipeds to do. That is why they would have a splayed toe for helping to grab onto the trunk of the tree. Artie has small canines unlike chimps, and this is another key defining anatomical trait of hominids. Large canines are used to display and intimidate rivals and fight predators. Their small size may suggest that within group, violent confrontation for access to females may have stopped. Importantly, there are no stone tools discovered associated with Artie, at least not yet. So with the Ardipithecine stage, we see the appearance of two key traits that we recognize as defining traits for the hominin lineage, bipedality and reduced canine. But we do not see clear evidence for those key unique features of humans, advanced cognition and prosociality. And in fact, advanced cognition does not seem to be expressed at all among this grade of hominids. However, the bipedality adaptation may open the door or operate as a pre-adaptation for some interesting things to come. By freeing the hands, mothers can carry babies, allowing prolonged periods of child dependence, which is key to the switching on of an emphasis for learned behavior. It also frees the hands for using tools. The reduced canine is often interpreted to indicate less aggressive male-male competition. Is this a sign of greater social cohesion and intergroup cooperation? While Ardipithecus now provides an excellent and earlier record for bipedality, it was Australopithecus afarensis and Lucy who first broke new ground in our understanding of the origins and time depth of bipedality. Lucy was discovered by ASU professor and founding director of the Institute of Human Origins, Don Johansen, based here at ASU. He is shown here to the left in a red shirt, with me to the right, 10 years younger. Lucy and fossils associated in the same sediments provided a nearly complete hip and well-preserved femora that clearly showed committed bipedality by 3 million years ago. When Lucy was found, there was still debate over whether enhanced brain size or bipedality came first. Lucy showed conclusively that bipedality predated increased brain size. If anyone doubted bipedality from Lucy, the finds at Laetoli put that doubt to rest. Here, about 3.5 million years ago, a volcano erupted and dropped volcanic ash across the landscape. It was rained on and then hardened in the sun after many of the animals that lived there walked across and left their footprints and trails, antelope, carnivores, snakes, and several early hominins. Those footprints show no handprints. They walked entirely on 2 feet. Australopithecus afarensis dates to between about 3.5 and 3 million years ago. We have a large and outstanding fossil sample of many specimens and individuals, and this allows scientists to examine within-species variation, such as sexual dimorphism. This shows that these species were still highly dimorphic, males were much larger than females. So this suggests that either males were still competing for females, or males provided a large part of the defense against predators, or both. However, the canines are small, so this goes against the male-male competition interpretation. It is clearly a biped, but still retains some tree climbing features, but not of the extent of Ardipithecus. The brain is no bigger than a chimp, and the tools have not yet been found, though one controversial report suggests they used unmodified stone to crack bones and cut off meat. There is one fossil site where there are a large number of individuals that seem to have all been killed at once, suggesting they lived in groups, but this is not surprising. Coming back to our figure showing the general pattern of human origins, we see that between 4 and 2 million years ago, there is a radiation of Australopithecine species. Even more interesting, there is a new adapted type of Australopithecine that evolves, what we call the robust Australopithecines. Robust Australopithecines are just that. They have big, robust crania and teeth. The first one that was found had such large teeth it was called Nutcracker Man, and its skull is depicted here. There are 3 known species, and the lineage is found in eastern and southern Africa. They have very large, flat teeth with thick enamel, and giant muscle attachment areas on their skull that were there to anchor large muscles involved in mastication. They had small brains and were very sexually dimorphic. They eventually go extinct roughly about 800,000 years ago. One interesting implication is that their appearance and extinction suggests that bipedality and small canine does not inexorably lead to the modern human adaptation, dependent on advanced cognition and prosocial behavior. At the same time that robust Australopithecines are evolving, another lineage appears, and we call this early Homo, in recognition that they are grouped with our genus, Homo sapiens. They have several features that align them with us, and most important of these is larger brains. The brain size rises to between 600 to 800 cubic centimeters from the 400 to 500 cubic centimeter range it had been in since the earliest hominids. There are a variety of early Homo species, and right now there is disagreement among scientists as to how to classify them, what to name them, and how many species there are. Roughly coincident with the earliest dates for early Homo, we find the first evidence for the manufacture of stone tools, and this signals the earliest archaeological record. The earliest stone tools date between 2.6 to 2.5 million years ago and are found in Ethiopia. These stone tools belong to what we call the Oldowan stage, named after Olduvai Gorge, and they are very simple. Most of the time they are cobbles that have been flaked a few times, so the assemblages are composed of these flaked cobbles, flakes, and the debris from the flaking process. Here are some examples of some very early flake stone tools from the Hadar in Ethiopia, excavated by the Institute of Human Origins here at ASU. Notice that they are all river cobbles. This is fairly typical for these early stone tool assemblages. Studies of the sources of the stone for stone tool production show that, prior to 2 million years ago, the stone was always collected from sources that were very close by where they had been discarded, no more than a few kilometers away. This suggests that hominids used local stone and did not carry it very far. And here are some of the flakes that are found at these same 2.3 million-year-old sites in the Hadar. Notice that these flakes were never flaked again, or what we call retouched or reshaped. We think the flaking process was mostly designed to create sharp-edged stone for cutting. But what were they cutting? Even at this early period, we find the fossil bones of animals with surface modification attributable to hominids. This includes several types of surface modification, such as cut marks from cutting meat off the bone, and hammerstone percussion marks for breaking bones to access marrow. A food resource that early hominids were very interested in resides inside the bone in the form of marrow. This is an extremely energy-packed, fatty food item, and equipped with a hammerstone, hominids could crack these bones and access this food resource. The image to the left shows this fatty substance, and to the right, you can see the marks produced. We call these hammerstone percussion marks. Experimental work has allowed us to develop criteria that allow analysts to identify marks of this type correctly 95% of the time. Hominins were also interested in the meat on bones, and they used sharp-edged flakes to remove that meat, leaving cut marks on the bones like these. Both percussion marks and cut marks are found on fossil bones at this time, though they are very rare. One of the best studied and spectacular localities is Olduvai Gorge in northern Tanzania. Olduvai Gorge is a deep erosional cutting in the Serengeti Plains. You can see the gorge here cut into those plains. This is a photo taken from the edge of the gorge. At the base, the sediments date to nearly 1.8 million years ago, and at the top, they date to about 20,000 years ago, a very long sequence. You can see in this photo the many layers in the sequence. Much of the sediment is volcanic ash erupted from volcanoes that you can see in the background. This is the site called FLK Zinjanthropus, where the first robust Australopithecine skull was found. Specimens of early Homo were also found here, but the site is best known for the dense archaeological remains. Thousands of Oldowan stone tools were excavated by Marean in association with the bones of many species of animals, and many have stone tool cut marks on them, showing that the hominins were actively engaged in consumption of animal tissues. There is debate over whether or not the hominins were effective hunters of the large antelope, or were scavenging from carnivore kills. Mary Leakey did the excavations, and she plotted each of the finds in 2 dimensions, and you can see the resulting plot here. There is a clear concentration of flaked stone and fossil bone in this cluster, and some scientists have argued that this is a campsite, similar to what modern humans create. Sometime around 1.8 million years ago, we have evidence for the first out-of-Africa migration of hominins, and there is evidence that hominins may have spread all the way to Eastern Asia. The most spectacular early site outside of Africa is called Dmanisi, in the modern country of Georgia, in the Caucasus Mountains. Dmanisi is an unusual site for several reasons. First, there are several skulls in fantastically complete preservation. You can see an example in the photograph to the left. Second, there are stone tools present, but there are very few. A few are illustrated here at the base of the slide. And third, the concentration of fossils is found stratified below a medieval monastery, and there is a very dense concentration of fossil bones. The evidence shows us that between 2.6 million and 1.6 million years ago, we see the evolution of the foundations of our genus, the genus Homo. The hominin is fully bipedal, but early in this phase, there is still some evidence for the retention of some tree climbing abilities. The brain size has increased to roughly twice the size of chimps and is now between 800 and 1,000 cc's by 1.6 or 1.8 million years ago. The body size is still rather small. Stone tools appear and then become reasonably common. The tools are simple and made on raw materials that are mostly collected from nearby sources and stream channels. There is some exploitation of meat and marrow of animals, and for the first time, our hominid ancestors leave the African continent. At the beginning of this lecture, we discussed how modern humans are uniquely characterized by an extremely advanced cognition and highly prosocial behavior. Do we see evidence for either of these 2 characteristics in the record that I've just reviewed for you? Certainly with Australopithecines, there are no signs of either of those features. Australopithecines have small brains, so there is no advanced cognition. We did note that early on, Australopithecines show a reduced canine, so we might be seeing some evidence of reduced within-group aggressive behavior, though certainly it would be a stretch to argue that this suggests prosociality. With the appearance of early Homo, there is some evidence for a more advanced cognition in that the brain increases in size, and we see the first stone tools. This could be evidence for a commitment in the adaptation to solutions that rely on the brain, technology, and culture. This could be a tipping point in the evolutionary direction, where the emphasis on adaptation is with behavior and culture, and away from slow, genetically constrained changes in anatomy and behavior. This slide provides a word list of important terms and names and concepts that we learned in this lecture.